Well, hello and welcome to everyone joining us for this X-Talks webinar. Today's talk is entitled Optimizing Amorphous Solid Dispersions, Overcoming Scale-Up Challenges with Next-Generation Technologies. My name is Corey Stanton and I'll be your X-Talks host for this event. Today's presentation runs for approximately 60 minutes and the webinar includes an interactive Q&A session at the end. To make the most of this session, feel free to submit questions and comments for our speakers throughout the presentation and we'll try to attend to as many as possible. If you're joining us via GoToWebinar, you can use the questions chat box, which is located in the control panel on the right-hand side of your screen. And if you require any assistance, please don't hesitate to contact me by sending a message using this very same chat box. And if you're viewing this presentation from our LinkedIn Live event, you can submit your questions using the comments tab on LinkedIn. Please note that all audience members are in listen-only mode and that this event will be recorded and made available for streaming to those who registered on xtalks.com. At this point, I'd like to thank AustinPx, who developed the content for this presentation. AustinPx was founded to help developers realize the full potential of their drug candidates. There are many obstacles to bringing a drug to market, including poor bioavailability and accelerated timelines. That's where AustinPx comes in. Their client-centric approach is designed to simplify the complexity of outsourcing, and their team of formulation, analytical, and manufacturing experts work with you to overcome challenges and identify opportunities to do more faster. As the inventors of KinetiSol Technology, a next-generation amorphous solid dispersion technology for poorly soluble APIs, with its smaller footprint, broader design space, and greener processing solutions, AustinPx aims to disrupt the ASD industry. From poor solubility to accelerated timelines, we all know that drug development is inherently complex, and effective communication and flexible processes can help simplify your path to market. That's why AustinPx has developed a client-centric approach to contract development and manufacturing services. They are driven by their client success and intentional about differentiating themselves through flexibility and responsiveness, as well as consultative, unbiased, solutions-driven development services. Now, let's take a moment to meet our panelists for today's presentation. One of our speakers today is Dr. Justin Keane. Dr. Keane has over 20 years of experience in pharmaceutical formulation development, clinical manufacturing, and process validation. As SVP of Operations at AustinPx, he leads manufacturing, quality control, facilities, and IT teams. Previously, he worked as a formulation scientist at DPT Laboratories and PharmaForm, gaining experience in hot melt extrusion and clinical labeling. Dr. Keane holds a PhD in pharmaceutics from the University of Texas at Austin and has supported over 30 new chemical entity campaigns. And joining Dr. Keane today is TJ Highley. TJ has 25 years of experience in the pharmaceutical market, holding various operational and customer-facing roles. As the former owner and executive leader of Microsize Inc., he led business development and site quality, guiding the company through acquisitions by Capsugel in 2015 and Lonza in 2017. After buying back and rebranding the business as Microsize, TJ expanded capabilities in analytical characterization and manufacturing, including partnerships with AustinPx's KinetiSol Technology. And now, without any further ado, I'd like to hand the mic over to our first speaker, Dr. Keane. Justin, the floor is yours.
Thank you, Corey, for the introduction and for organizing the event today. I want to start by just kind of providing some background and perspective on amorphous solid dispersions. I think the audience is quite familiar with the term and the need for these types of enabled drug products for the market. But we have challenges when we try to bring these through the development process, and I think it's important that that we break down these challenges and look to the future at ways that we can overcome them. So to start with some background, there's a couple ways to produce amorphous solid dispersions. We can start with bottom-up processes, such as solvent evaporation-based methods that produce a ASD particle at the same time as ensuring that the amorphous polymorph is formed. Alternatively, we can use melt-based methods or top-down methods where a dispersion is produced and then a subsequent operation shapes it into its dosage form. And it's worth noting that these aren't the only approaches. There are other methods, but spray drying and hot melt extrusion are certainly the most relevant from a commercial product perspective. In this figure, we can look at this from a timeline perspective. Amorphous solid dispersions have been known in the art of formulation technologies since the 1970s. However, we saw a real surge in the mid-2000s in development due to advances in the way molecules, new chemical entities are developed. This has resulted in molecules that really need these enabled compositions in order to achieve relevant blood levels, achieve bioavailability targets so that they can be clinically effective. And we can see that spray drying and hot melt extrusion have really found a niche in solving these issues over the last 15 years. And there's a few other technologies sprinkled in, and we certainly are looking towards the future at what we would call third generation amorphous solid dispersions, and we expect to see KinetiSol amorphous solid dispersions approved in the near future. And then I'm really looking forward to revisiting this type of a timeline in the next 10 years. Now, regardless of the manufacturing approach, we can break down the process control strategy the way the industry does is, for reference here, the ICH Q8 document. What we want to start with is defining the quality target product profile, and then identifying which quality attributes could be deemed as critical. So the term CQA applies here, and then doing risk assessments, assessing what could cause these CQAs to not meet their targets. And in order to do that, we have to design and execute costly studies. This takes time. We need to understand the potential environmental, material, equipment, and other attributes or process parameters that may affect those quality attributes. And we can package all this together to develop a design space in support of the product control strategy. And as we move to looking at these enabled amorphous solid dispersions and bringing them through this process to the market, these types of strategies can become quite complicated. So when we look at an amorphous solid dispersion platform, so we're an example product from a CQA standpoint, there's certainly factors that bubble to the surface as the ones that would be most relevant to this type of process. Any product needs to be manufacturable. It needs to have the appropriate densities and compressibility parameters for producing a final dosage form. There may be homogeneity issues or other potency concerns. These are all standard types of attributes that one would look at for any drug product. However, for an amorphous dispersion, the point is to control the polymorph, and this is what affects the pharmacokinetic exposure. And so this crystallinity parameter, or the lack of crystallinity, tends to be extremely relevant, but not just the crystallinity. As we move into this next generation of amorphous solid dispersions, or ASDs, we've really come to realize that the particle size distribution and the morphology of the particles that are produced can really affect this dissolution rate and also have a significant impact on total bioavailability. So we would always kind of expect a CQA to come from these parameters, and depending on the process, whether or not it uses heat, we have potential CQAs for process degradants. So if we zoom out from these details, every product needs to go from some lab to production with the potential for a site change. And with amorphous solid dispersions, we typically see that a product would transition from 1 to 2 or even 3 sites during its development cycle. In order to demonstrate control and get to this end goal of an effective design strategy for a product, we have to really consider our goals from a few different perspectives. The first, I would say, from a technical perspective, what are the theoretical fundamental mechanisms at play with the technology employed for the product? What can you model the process? Can you match the geometry as you scale the process as the clinical supply demand increases? Can you conduct the experiments in parallel with clinical supply processing? And what practical experience can we bring to bear for a particular product? Another approach to viewing, a scale up and tech transfer plan is from a regulatory and a documentation standpoint. You know, doing risk assessments early and often to understand knowledge gaps, identify potential process analytical technology needs are important. And as we look at scale up and transfers, you know, we should document what we learn, what we expect to achieve during a particular event through protocols. So there's the need to capture what we believe to be the future CQAs and report on that along this process. And then, you know, there's a more strategic view, especially if you're a large company and you have a portfolio to manage, is making sure that you have the right partners for the right technologies that you're employing. For example, if you can minimize capital expenditure and environmental impact, the total cost of your product development process may go down, and selecting partners that have existing infrastructure as well can minimize overall cost and speed to market. But what I really want to focus on for this topic is simplifying the process, you know, attempting to avoid simultaneous, highly coupled unit operations. And this is where we find the ASD space is challenged. So for example, when we employ a spray drying process to develop an amorphous solid dispersion, process engineers and scientists and manufacturing staff have to simultaneously demonstrate control of the heat transport processes that are happening as droplets are flowing through the spray dryer. And so this is a mass transport event that's happening. In addition, there's a mass transport event happening upon droplet formation and the evaporation of the solvent. And then all of these particles are flowing through to a separator, and there often are significant dependencies in these process parameters where optimizing one step in this, if you view particle or droplet generation as a unit operation, and then heating and drying as a unit operation, and separating as a unit operation, optimizing one of these may impact the downstream significantly, and often compromises or optimums have to be sought. Ideally, we don't form the particle that affects the dissolution kinetics at the same time that we produce the amorphous form. Hot melt extrusion is an example of being able to do that. However, what it gives up in terms of being able to separate particle size control from amorphous solid dispersion crystallinity control, it deals with in momentum transfer. So we still have these simultaneous heating events, mass transport events, but we also have to make sure the material's moving through the system at the correct velocities in the right places to ensure the process is in a state of control. And this is where KinetiSol really enters this paradigm and provides an alternative that's simpler. KinetiSol is a thermokinetic compounding technique that utilizes ultra high speed mixing elements that operate at extremely high speeds, producing significant amounts of shear. This is a solvent-free process. This shearing and frictional heating that occurs when these blades impart energy into the product produce the heat directly and allow for the material to rapidly heat up and produce an amorphous dispersion. And you can see from the inset graph that this would occur, you know, in about 10 to 15 seconds, depending on the process or the product. And most of that time is in initial induction phase. The heating that occurs is very limited. But what's really relevant from a product supply chain and an experimental planning and a total cost of development is that this dispersion formation is done as an isolated unit operation. Contrary to melt extrusion, there's no momentum constraints. In addition, you know, contrary to spray drying, particle formation is a separate unit operation. So all of the parameters that we would want to optimize for the best product can be controlled separately. This isn't the only benefit of applying an advanced dispersion technique like KinetiSol. There are inherent particle morphology benefits. In one of the earlier slides, I listed potential CQAs as manufacturability, compressibility. We've demonstrated through a variety of studies that KinetiSol processed ASDs, due to the nature of the mixing and the quenching process, produces dense and good flowing particles, but there is some residual porosity that provides excellent compression properties. And so this allows us to make a denser tablet with more drug, a higher payload per se, so that we can reduce the total pill burden and total tablet size that the patient ultimately sees. In other perspectives, from a supply chain or facilities capital perspective, KinetiSol is very limited facility requirements or overhead. The technology at commercial scale is modular, it's mobile, and it can fit in a typical processing room where one might find a tablet press, for example. And in the inset on the right, we can see this KinetiSol machine that produces a product at 10 to 20 kilos per hour compared to an equivalent spray drying instance on the left with an operator for scale. And we can kind of really zoom out from that and see that the benefit isn't just the size of the facility that one needs to have for a commercial machine. It's the fact that there's not multiple steps in order to iterate a product from development scale, for example, in a Buki up to pilot scale and into a large commercial instance. There's only 2 scales for KinetiSol, and we have different names for those. The KinetiSol formulator is the scale at which we develop formulations, and also the scale at which we can produce small scale clinical supplies. A shot, which I'll talk about a little bit more detail later, is about 5 to 20 grams. And when we manually process these shots on a formulator, we can achieve production rates anywhere from 5 to 200 grams an hour, which is more than suitable for a phase 1 clinical trial. And then there's the KinetiSol compounder. It's our larger scale equipment. It's got geometric similarity to the formulator. However, we don't need a third step to go from a phase 2 to a commercial scale. We can do that all with this machine. And it's got PAT fully integrated. The shot size is about 80 to 300 grams versus the formulator, and the production rates are up to 20 kilograms an hour. So just to put that in a more visual context, the KinetiSol formulator in the top inset is what we utilize during formulation development. And then we can rapidly progress a prototype into a phase 1 clinical trial example through the same scale transfer. So the process parameters, the equipment that was used to develop the formulation can be utilized directly in a quick transfer to cGMP operations for proof of concept. Once a phase 1 formulation has been identified as relevant from a bioavailability standpoint, that's when we do a step change, this single geometry matching scale event to the compounder, and then we start looking to a mode change and optimizing throughput as development continues. So to add a little color to the KinetiSol mode, we really are just talking about manually processing material through the compounding chamber, which we would call individual shots, versus a continuous mode where we've optimized the feed through some type of potentially co-processing. If blending directly doesn't meet the need, we may blend it, co-mill it, granulate it, if there's a need to for the particular product, dry it. And then the KinetiSol machine can automatically move the material through the system. And as I mentioned earlier, the bulk dispersion is controlled by the compounder, but the particle morphology and the final dissolution rates are controlled through milling, which is one of the reasons why we've partnered with Microsize, and they can talk much more about that later. There is a question that comes up quite routinely when we start talking about the semi-batch or the continuous nature of a KinetiSol machine. The benefit, as I mentioned earlier, is that we can separate this compounding unit operation from the particle development unit operation, and we can isolate the process parameters from other unit operations that are happening, and we do that by isolating and running the process on small amounts of material. KinetiSol is not the first process to do this. The example that I think best embodies a regulatory consideration for how the process works is a granulation by slugging. And this is a machine that does a continuous set of coupled unit operations. The process aliquots out some volume of material, transfers it in the machine, and then applies a compressive force to granulate a formulation. That material is then transferred to an ejection location where it's discharged from the machine. And so a tablet press does this continuously, and we do this by feeding a powder to the machine, and we get what's termed a slug out of the machine. And these slugs can be milled into a powder and subsequently turned into a final dosage form. So this is a well-known approach. KinetiSol does the equivalent to this, but instead of compressing a slug, after we've aliquoted the material and transferred it into the compounding chamber, we apply a shearing force, which melt granulates the material, and then we transfer it to a quenching section and discharge the material. So the term of art here versus slugs is shots. So we take the continuous KinetiSol compounder, takes powder in and creates shots of material. Just to step into that a little bit more, the orange parts of this image on the left are the product contact pieces of a compounder, and just going to step through how this aliquoting to discharging operation works for the audience. So the first thing that occurs is an aliquot is weighed out using a gravimetric feeder. This feeder, is on a scale, so we can set the exact weight that we need for a given shot, and that material is put into what we call a transfer tube. When the machine is ready to process this shot in the compounder and chamber and make it amorphous, this actuator opens, and another actuator transfers the material into the chamber. This is the transfer tube. And then once in the chamber, the process that I described earlier happens. The mixing elements rapidly increase to the target RPM setting, and a PAT probe looks for process completion. Once that has been achieved, the material is ejected and then cooled by smashing and quenching between two plates where it is discharged. So with that background, we can talk a little bit more about the process parameters that are relevant to a KinetiSol ASD. The first and typically most common type of scale-up evaluation that's done for any product is really understanding the powder blends and potential handling concerns, understanding potential for segregation, making sure the powder can flow, understanding if there's a friction issue that needs to be managed with any types of hoppers that are employed. This is a standard scale-up issue for any solid oral dosage form, and we understand this as an industry. We need to manage vessel sizes, we need to mix to create blends, we need to understand the particle size distribution of our supply chain, and make sure the material has the flowability and density parameters that are necessary for it to flow into the system. And if the formulation requires some physical adjustment in order to ensure this, then we make those changes. Once the blend is in the compounder, we have a very strong understanding of the theoretical considerations and the practical experience to ensure control. I think there's a lot of equations on the screen. I don't want to get bogged down in any of the kind of physics lessons here, but at a high level, we understand that as we transfer a process from one machine or one facility, we need to understand that there are some energy parameters that may change. For example, the environment may be slightly cooler, the humidity in the room may be slightly higher, and that the coding models that have been developed really can teach us how to manage this. For every type of thermodynamic change to the system, there's always something that we can use to counterbalance it. So there's a bunch of parameters that are controlled basically by the machine, and then there's some cooling parameters that we can adjust to make sure the process is successful. But the real part of controlling the process is it's a mixer, and it has a rotational speed. We know that we're going to do a single scale-up event, and we can apply a power number, tip speed relationship to ensure that the RPMs are sufficient to achieve drug dissolution, which is also significantly impacted by the temperature at which we process the material to. We know that quite a few melt extrusion and certainly most KinetiSol processes utilize a dissolution process where the process heats up the carrier polymer, it softens and becomes a solvent for the drug, which dissolves into that. And we know that the temperature and the mixing rate or the shear rates can significantly impact the duration required for that. And the counter to this is the longer we heat up any system, the more potential for drug degradation. So we can look at all of these relationships and really sort of simplify this. And again, keeping in mind that there's no momentum balancing here. There's no particle development at this point. What we're really looking at is fixing the shot mass, because that's a control parameter set at the feeder, and then understanding the environment and making sure that the machine is operating in a place where the temperature is reset between shots. And we know how to control that from a very practical standpoint. And what this leaves is finding a design space where we know all of the crystalline input material has been rendered amorphous and none of the material has been degraded beyond any potential specifications. And because of the isolated nature of the process and the fact that we can produce a shot of material, it's extremely practical and fast and cost efficient to map the temperature and speed space that renders the quality attributes in control. And we've done this in practice. We are looking forward to commercializing products. We've established registration scale processes where we've not only demonstrated these process maps in terms of temperatures and speeds and their potential impact on certain drugs that may be thermally sensitive, but also we have the ability to not only use temperature as a way to control or monitor the process, but we can take a Raman spectrometer, for example, to the line to conduct process development experiments or collect data during registration and validation batches to support the final process control strategy. So in summary, KinetiSol itself has shifted the ASD boundaries in enabling streamlined scale up and manufacturing, allowing us to significantly faster process a material not only in the chamber and render products amorphous, but through the development cycle. And that's been the focus of this piece. But beyond that, it's a broader solution that we've found is applicable to a wider range of molecules. We have a broader design space in terms of the number of excipients and types of chemistries that we can deploy to make a formulation the best it can be for a drug. But it's also greener, and from a capital, operational perspective is much simpler and cheaper. And beyond that, there is some extended potential for IP to protect products through their life cycle. And so with that, I want to turn it over to TJ to talk a little bit more about particle size control. as we have demonstrated that KinetiSol allows us to produce a dispersion, and then it's a separate optimization example to produce the right particle for a product. And being able to decouple that and bring an expert like Microsize to the table is very beneficial.
Great, thank you, Justin. So where Justin left off with the compounder and the ASD formation, what comes out of that process is a stable, solid form factor that's about a couple inches in diameter, very thin, very brittle, and that form factor needs to be converted into something usable, and that's where the particle size control and morphology control optimization opportunity comes in. So the first step would be to take that pancake shot that Justin described and turn that into a powder using just a simple delumper or jawbreaker type milling operation to get you out of a solid and into a diverse PSD powder. Two things with this is a delumper in that first step gets you a very wide distribution of particle sizes, anywhere from millimeters down to hundreds of microns. And if you can look at the morphology of the particles there's two things going on there. There's dissolution coming from the surface of the particle, but also a tremendous amount of pore volume that acts differently than the surface in terms of surface area and dissolution. So getting that ratio of surface to pore volume dissolution optimized is another opportunity for adding additional particle engineering. So with that, that coarse milling has opportunities for further particle size engineering and control. Microsize has a range of very deep technologies in particle size reduction. So every milling technology that you could think of, either mechanical milling or air jet milling or fluid bed milling, is available. So hammer milling to get you into a finer powder than the delumper, pin milling to start to move to a more homogeneous shear environment and particle size distribution curve. If the excipients that were chosen have heat problems, we could move into an ambient milling process with either loop milling or spiral jet milling. So using compressed gas to create the motion and the milling energy to cause particle size reduction, and a very homogeneous environment giving you a Gaussian distribution curve. Additionally, if the material does not want to, if the target formulation doesn't want to be too fine, we can use classification technologies as well and pull out the particles from the delumper that are objectionable. So remove fines or remove oversize if the bulk particle or the majority of the particles are in the range desired for that particular use case. So we will pick the distribution and technology based on the target particle size distribution, the desired dissolution with these two different surface areas dissolving over time, and optimize that. And then also understand the physical properties of that compounded powder and that excipient choice and that API molecule, understanding its physical properties and flow through a particular mill to optimize it and make sure that the milling process is problem-free. So with any milling process, it's pretty straightforward scale up. It's balancing the feed rate of the process with the milling energy. So that milling energy can, if mechanical mill is the rotor speed and the power factor, and then the tip speed specifically, depending on which scale mill we're talking about, matching that as we scale up from early phase to commercial. And if it's a fluid jet mill, it's balancing the grinding pressure that's creating that milling energy. So either one of those becomes a CPP for us. and as well as balancing the material that energy is being applied to in terms of feed rate. So that feed rate is definitely influenced by holding constant and optimizing which feeder we select, which screw configuration is present if it is a screw feeder. And with both of those, we can evaluate vibratory feeders, screw feeders, either single screw or dual screw, volumetric feeding versus gravimetric feeding. So all of those configurations are possible. Decide on which one is best for that particular DPI, hold that constant, and then explore the feed rate, lows and highs that are possible throughout development. And then additionally, mill configuration, as we talked about those different technologies. Once the technology is selected, then optimizing and holding then constant that configuration. And that can be certainly mill size and chamber volume and nozzle angle, but it may even be as minute as things of the venturi that helps feed the material into a given jet mill, or the screen size that's present in a mechanical mill. So the CQAs in a milling process that we're trying to control and keep constant are the particle size distribution that's wanted, and then that leads to the dissolution that's present. So what we have found in any scale up factor is definitely understand the entire PSD curve and be tracking all points, namely the bottom side and the top side, the D10 and D90, and the median D50. And so when we develop a process, we tend to, depending on the distribution that we're talking about, develop that process and control it to the most difficult to control channel, and the others will fall in line. So if oversize is the issue or large particles is the CQA issue that we're trying to control, then we'll control to a D90, but still track a DV50 and a DV10 to understand what those are. But more typically, we find very Gaussian distributions in our processes. They're very fine and small in breadth. So we'll control to a D50, but just some additional tracking and targets around a D10, D50, and D90, but not a spec. Secondly, for the dissolution, we have an added capability and that we'll bring in early and look at the dissolution not by chemical means and shaker flask and HPLC at different points in time through a dissolution experiment, but actually had imaging using a SPA technique developed by the solubility company that's on-prem at Microsize. So there we can watch every second of dissolution behavior, and where it's extremely relevant is we can make that dissolution experiment in aqueous phase or biorelevant media systems. We can watch every second, and every polymer system and ratio of API to polymer, depending on those properties and the polymer selected, as that dissolves and that surface area dissolution versus pore size dissolution, that can be watched every second. We can watch polymers swell or gel or linearly dissolve, as one would expect, but really use that knowledge to characterize and drive the formulation choices. And so our scale up process is typically a three-step process where we'll do feasibility and then understand that empirically what ranges work and what technology can give that desired particle size distribution, and then understand in that first event the material's flowability and handling, and then be able, and cleanability, and be able to solve those things for future events. We have the ability to add vibration or modify the equipment in terms of adding air assist or pull additional CFM to help any sort of handling or flowability issue. We can change venturi angles and have all sorts of handles to adjust. And usually that's demonstrated during the feasibility event, and we come up with a good plan for the qualification batch to optimize that. Yeah, the qualification batch is typically can be either continuation of empirical design using the practical experience over 30 years, or doing a full DOE assessment if material allows, and getting contour plots developed for the CMC package. And to that particle size and dissolution, there's always the ability to confirm the crystallography, or the crystallinity rather, understand the rheology of what we're making, and do all of that in-house. And then once that DOE is done and the edges are well defined, we can then move on into validation and understand the overall process variability by running several batches and having an aggressive sample plan to look at the overall variability and of the CPPs, holding at steady state, pushing rate, understanding the min and max and average, and standard deviation around the particle size changes with a steady state process, and coming up with the overall CPK. So with that, that's the technical piece of the milling, and the scale up is to map those two out and understand that design space. But we have found with Austin, the qualitative aspects of this tech transfer is really the key. So and that starts with the qualitative aspects of having a solid relationship. So both companies understand from a leadership perspective, from analytical and benchtop and process engineering, all the contacts know each other, deep appreciation of each other's strengths and weaknesses, and that allows already cross-sharing to occur and recommendation of what optimization to pursue and how each other's facilities can help. There is an established plan for MSAs, and quality agreements are in place, and CDAs. All of that is already onboarded to benefit the client and reduce the overall burden of scale up. The transfer packages of the compounding into Microsize for GMP commercial production are aligned in terms of batch record format, what we need for a compound onboarding, the test method transfer, all of that's been ironed out. So not every project needs to restart from ground zero. The share protocols are also present in terms of how we share those documents, and creating a collaborative space IT-wise to do that, creating a secure transfer of compounder data that comes from the compounder directly transmitting to Austin so they can assess every run, every batch. And we have a plan to do man and plant shadowing from a very early level. So during phase 3 optimization, there will be great communication between both parties, and be shadowing from an area as Justin designs that space and does those trials. Microsize engineers and formulators will be present to streamline that transfer to our facility. And then, as you expect, a like-for-like compounder between facilities, overlapping analytical capabilities, both solid state and PSD or laser diffraction. And then in the possibility of early transfer of those TMs as well during phase 1 or phase 2 is possible, so that those are all ready to go and onboarded prior to phase 3 transfer. So with that, I'll transfer back to Corey for Q&A.
Excellent. Thank you very much for that insightful presentation. And as TJ mentioned, we are now ready to begin the Q&A portion of the event. And as I do so, I invite the audience to continue sending their questions or comments. Just a reminder, if you're attending via GoToWebinar, please use the questions chat box. And for those of you watching on LinkedIn Live events, feel free to post your questions using the comments tab. We already have received some questions here from the audience, and we'll get through as many as we can with the time that we have left. But rest assured, there will be some contact information that you can use to reach out to the team. So if we don't get to your question, there will be a chance for follow-up after the webinar. First question that's come in here from the audience. This audience member is asking about what is the typical loading of API to dispersing polymer? Is this different from what might be used for a conventional ASD?
Thanks, Corey. Yeah, I really like this question, especially the fact that it's two parts, because the short answer to the first question is it's typical drug loadings up to 60%. Our lead clinical asset is 50%. But what we really found that KinetiSol is best at doing is producing a lower drug-loaded dispersion that has the right particle properties to drive the dissolution performance that's optimal for the product. We've seen multiple examples where a lower drug load allows us to deliver a lower dose to the patient, achieve the same bioavailability, but at the same time, make sure that the payload is equivalent or smaller than what would be produced, say, from like a typical 50% spray dried dispersion. So what KinetiSol does that's different is it allows a higher performing particle that can produce a denser payload with higher bioavailability.
Great, excellent. Thank you for that. Another question that's come in here from the audience. This audience member was asking about product contamination. So the question was, about how do you lower that risk between two sites?
I can help with that. So primarily that's just rigorous cleaning protocols. So the deep understanding of how to disassemble, reassemble, and break down that equipment. So every square inch is addressed and not only cleaned, but then visually inspected and then able to be swabbed for chemical residual. We test using HPLC for that cleaning verification. And then it comes down to suite design and general procedures around the production operator. So these are all done in segregated suites with dedicated HVACs and multiple layers of HEPA filtration, airlock control, and then just general housekeeping is primarily how it's controlled.
Great, excellent. I'll keep it with you, TJ, because I think this next question is probably best, directed your way. This audience member was asking about Microsize's experience with scale up for pharmaceutical commercialization.
Great question. So yeah, our experience is being a commercial facility. 70% of our production is commercial approved products feeding worldwide markets. So regulatory inspected by US and worldwide regulatory agencies and supplying commercial product on a routine basis. So certainly the technologies that we have from particle size reduction are routinely scaled up here, but we've hosted other technologies, including a nanosizing one, and then now the Austin compounding ASD formulation. So we'll just apply that tech transfer plan, document that all here, and scale up in collaboration with the client.
Great, excellent. And kind of on that topic, we just had a question come in wondering about just if you could kind of boil it down to, one piece of advice you would give to a company, preparing to tech transfer a product, for scale up and commercialization. What would be that, main piece of advice you'd go with?
I think it's establishing that trust early and understanding that the strengths and weaknesses of both organizations that would be involved in this ASD and how they can interplay. And I think really a push to understand their use cases will help develop that trust and understand the diversity of challenges that they've addressed, whether it be bioavailability or reducing the load or the form factor.
Great, excellent. I think we have time for one more question here from the audience. This audience member was asking about, with the tech transfer and a scale up of KinetiSol from AustinPx over to Microsize, whom do you envision being responsible for the final product release, and what would be the typical COA tests for a final release?
Justin, I'll let you take that one if you could.
I apologize, my microphone blipped right at the tail end of that question. Could you repeat it?
Sure, absolutely. Yeah, and I think this, as I was saying, this will be, I think, our last one that we'll have time for. With the tech transfer and scale up of KinetiSol from AustinPx to Microsize, whom do you envision being responsible for the final product release? And then what would be the typical COA tests for the final release?
Yeah, in terms of the tests, I think, risk assessments and the process that we go through in establishing the CQAs will define, what's critical to test before a dispersion goes to a commodity CMO to get turned into tablets. So, typically you would see a particle size distribution confirmation because that's core to the dissolution process. You want to make sure that the batch is indeed amorphous. And because we are applying a thermal, process here, we would kind of look at assay impurities just to make sure that the product is ready to utilize at the final site of manufacture. In terms of who can conduct that test, both AustinPx and Microsize have a variety of analytical capabilities, but also the site that's going to be accepting the tablets and responsible for the final product chemistry can play a role here as well, or any third-party lab. So really comes down to the supply chain and the risk analysis.
Okay, well, great. Thank you both so much for those answers. Great questions during our Q&A session. At this point, we have reached the end of our session. And as I mentioned earlier, though, to our audience members, if we couldn't attend to your questions, if you have any further questions, or if you're someone who's watching the recording of this webinar, you can always get in touch with the team at AustinPx using the contact info up on screen. There's an email address and the web page as well, and they'll have a chance to follow up with you after the webinar. We do thank everyone once again for participating today. If you're attending via GoToWebinar, I have added a link to the chat box where you'll be able to view the recording of this event. And you can, of course, share this link with your colleagues so that they may register to view it as well. You'll also be receiving a follow-up email from XTalks with access to the recording. And after we wrap things up here, a survey window will appear on your screen. Your participation in the survey is always appreciated as it helps us improve our webinars. For those of you watching on LinkedIn Live, we do encourage you to create an account on xtalks.com and register for this event, which will, of course, then give you access to today's recording. Now, please join me in thanking our speakers once more for their excellent presentation. Justin, TJ, really appreciate your time. We hope our audience found this insightful and informative, and we wish you all a great rest of your day. Take care, everyone.
Thanks, Corey.
Thank you.